Welcome everybody. It's my pleasure to begin our webinar, the first of the series, Toward New Horizons, the Hebrew University experts share their insights. Next webinar will be on Thursday, where we will speak about climate science in the time of Corona. Today we have the honor to host Professor Alexander Rouvinski, a researcher at the Institute for Medical Research, Israel, Canada, at the Hebrew University's Faculty of Medicine. He will give a lecture on immunotherapy as a strategy to treat acute coronavirus infection. Professor Rouvinski is an expert in microbiology and molecular genetics. He investigates how viruses originating in animals can evolve into human viral pathogens. His research focuses on the new and infectious viruses spread by insects that are a major causes for human and animal diseases worldwide. After his lecture, you're welcome to ask questions by raising your digital hands. And now, Professor Rouvinski, the floor is yours.
Hello, thank you for joining this webinar. Actually, I represent what we call a team CAREM, and I will explain in a moment. And what I will tell you about is the immunotherapy as the strategy to treat acute coronavirus infections. Here you see Team CAREM, which is a consortium of many labs here in our campus and also other labs in the Hebrew University. And we are CAREM team because we are situated in the NCAREM campus, and here you can see the Mary Spring in the NCAREM village down the campus. And here you see we are working on the coronavirus with the masks, and here you see a picture of us 100 years back. Of course, I'm joking. This is the flu pandemics, Spanish flu pandemics, and you can see some similarities. So I will just speak a few words about the viruses in general. So viruses, as you may know, they are obligate parasites of their hosts. They cannot replicate on their own. They always need the host, or in particular host cells, living cells, to propagate. Often they cause a disease, but not necessarily. So they are in a way not alive, so they don't have their own metabolic machinery, but they do sense the environment. And I would say they have two different entities in their life cycle. So one is being a virion outside the host cell, and another is being getting a life inside the host cell and propagating and subverting the host cell machinery, the process that often causes the disease, to replicate their own genome and their own particles. So they are really invasive and subvert their hosts. So they cause, many of the viruses cause transient infections. So they multiply in the host and then they leave the host often, not all the viruses, but those we are speaking on today. And then they want to propagate from one host to another. And indeed, some viruses, they propagate only from human to human hosts, while others have a so-called animal reservoir, and they start from animals or even continuously propagate within animals and then can go back into the human, so-called zoonotic viruses. And how can we break this chain of host-to-host spread? So, of course, currently we know the strategy of social distancing. Vaccination can spread, but not always. I will tell about that later. And of course, so-called herd immunity. Okay, so I'll go a bit deeper into the coronaviruses. So coronaviruses are large RNA, ribonucleic acid viruses that cause diseases in mammals and birds. And they include several human respiratory viruses causing several human respiratory diseases. So SARS-CoV-2, which causes the COVID-19 disease. And here I would just like to explain that in a way, viruses not always cause the disease, and we differentiate between the disease and the viral entity, as we will see in a moment. So I will just mention a few previous pandemic outbreaks of coronaviruses, SARS-CoV-1, SARS-CoV, which happened in 2002 till 2004 and caused severe acute respiratory syndrome. MERS, which is a Middle East respiratory syndrome, and it originates from camels, and of course the recent pandemics of SARS-CoV-2. We also know so-called common cold human coronaviruses that are in a way endemic in a population and quite widespread. They often are not associated with severe diseases, but they are very common in the population around the world, and here we list some of them. So this is the phylogenetic relationship, or in a way their family tree. You can see here that this so-called beta lineage B coronaviruses include SARS-CoV and SARS-CoV-2, the recent outbreak. And here I indicated their animal hosts as well. And we can also see this so-called alpha coronaviruses and beta coronaviruses from different lineages, which are closer to each other than those of alpha, but they are still distinct. And I would just like to mention so-called human coronavirus OC43, which has its very close species constitutively infecting bovine. And also there are coronaviruses from pigs, and as I mentioned before, coronaviruses of birds. So just a few words about the genome organization of coronaviruses. The genome of coronaviruses is really large. For RNA viruses, it goes to 30 kilobase, and it's a positive sense viruses, which means that they actually encode directly for the proteins. And they have multiple open reading frames. And to maintain such large genome, they have very atypical feature for RNA viruses. They have a high fidelity in replication, so they have this so-called proofreading polymerases, but they are known to undergo recombination during the replication process, which may generate new species. And it is quite challenged to understand the way they package their genome. They assemble the particles, mature, and how they leave the host cell. And we are studying these processes now, and this would help us to understand also how to generate reporter particles, as I will mention in a moment, virus-like particles, and reproduce complex three-dimensional antigens. So just a few words about so-called structural proteins of the virus. And when we say structural proteins, it exactly points to the subject I mentioned earlier. Here you can see a virus particle. This is the form of the virus life, I would say, cycle, which is outside the host cell. So it is not metabolizing particle. And so all the genes that comprise these particles, or the proteins, they are called structural, those that are building the particle. So we have here this so-called spike of the coronavirus. And indeed, this spike is bulging out of the surface, as you can see here, of the virus. And this is why we call this virus coronaviruses, because they remind in projection a crown, you see, like corona crown. And this is the most important viral determinant for the entry, but there are more, as you will see, there are more antigenic determinants. So what is our approach? Our approach relates to immunity. So we would like first to understand the immunity. So immunity can be, in a way, divided into the innate and acquired immunity. And immunity is usually, of course, protective. And the whole strategy of vaccination is often very successful to provide the protection, to train the immune system. But occasionally there are rare cases where the immunity itself during the course of infection can be destructive. And this is, of course, in case of overreaction, for instance, cytokine storm. So sometimes even after the viral infection is resolved, the body continues to overreact, and this can cause an overproduction of cytokines and eventually very severe conditions. So the balanced immune response is really the key for successful resolution of the viral infection or infections in general. Another instance where the immunity can be deleterious in a way or destructive is a case of so-called antigenic sin. And I would like to spend a few minutes just explaining the concept. So in case of antigenic sin, the pre-existing immunity to a similar entity, but not the identical one, can cause the bias of immune system and can drive a biased and deleterious immune response. But it's not always the case, but it can be in some cases. So for instance, in some cases, immunity against something similar is actually good. As we know from the case of vaccinia, you know, the word vaccine comes from vaccinia, and vaccinia virus, which is related to smallpox, gives good protection against smallpox. So this is a successful case of vaccinating with something similar, but not identical. In some cases though, like dengue disease, vaccinating with similar things, but not identical, drives the immune system wrong way, like misleads the immune system, and this is called antigenic sin. And there are instances related to coronavirus infections of cats that reminds this type of antigenic sin. So development of vaccines against coronaviruses should be well monitored because of this history, but there are also successful cases and recent reports. So just it needs to be well monitored and checked. So usually we can divide the immune response into the humoral arm, the development of antibodies, and cellular arm. Today I will focus on the humoral arm and discuss the antibodies which are found in the serum of patients. So antibodies are usually clonal. So there is an assortment of different B cells in our body that generate specific antibodies, clonal particular antibodies, but in the serum, all these B cells, they maturate and they secrete an assortment of monoclonal antibodies, which you can see schematically here. And so this mixture is called polyclonal mixture when each antibody attacks the viral infection from a different angle in a way. So really, the virus is a dynamic entity, so it always replicates and undergoes in a way evolution. And the constant pressure placed by the immune system to the virus is an important parameter. And of course, the antibodies are always maturating and mutating. And so it's always the race between the virus and the immune system. In particular, we are talking now about the antibodies. And the concept is to boost the humoral immunity. So why currently humoral immunity? Because antibodies are soluble and it's easier to work with them in a way of treatment. And this approach was pioneered by Pasteur in the 19th century toward rabies virus infection. And it often works. An approach called passive immunization that we will go deeper in a moment. But not always there is a humoral response in the natural infection. So for some viruses, there is no good humoral response which is raised. So antibodies can be produced in animals, in large animals, and they can be used as a passive antibody transfer, but this is not always trivial, and this can cause so-called serum sickness. But this passive immunization from animal sera is used since many years back, but it is still applied for several bacterial toxins, for instance, diphtheria toxin and tetanus. Of course, nowadays there are newer approaches like genetically humanized animals, those that express and produce antibodies that are human-like. So these animals can be vaccinated, starting from mice that was recently published just last week, anti-SARS-CoV-2 antibody from mice, humanized mice. And of course, going even to the cows, there are transgenic cows that express humanized antibodies. Those can be vaccinated and generate large portions of sera. Of course, during pandemic, we can go also to the donors, human donors, those people who recovered from the disease and are convalescent and contain large amount of antibodies in their serum. From there, we can also isolate the B cells produced generated in our body, and then using recombinant DNA technologies, we can isolate the genes encoding for potent neutralizing, protecting antibodies. And this type of approach is in a wide medical application in the recent years. It is even used for cancer treatments. So this, in a way, passive immunization is going beyond infectious agents and is applicable for many aspects of modern medicine. And the prerequisite for all this is really identifying the right antibodies in serum and then subcloning the right genes encoding for these protective antibodies from the B cells. And when we have an assortment or a cocktail of such antibodies, we can even use them as a cocktail. I would draw this parallel like antibiotics. So we can use one antibiotic, but often like a cocktail of antibiotics can give a solution. So in this way, we also think that the cocktails of antibodies or chimera between different antibodies can give better solution. Really, our first goal is to understand the immune determinants of the SARS-CoV-2, because we believe that understanding first can really help taking a rational approach to design the proper immunotherapy, rational and in a way personalized approach, because antibodies are targeting different regions of the virus, and the importance of these regions is different for the maintaining the life cycle of the virus. So in a way, if we look on the virus as a puzzle, puzzle composed of different proteins and different pieces, I want you to remember and to think on the fact that this puzzle is really three-dimensional, and the three-dimensionality of the structure of the virus is essential for its complexity and for its function. And so we really view the viral antigens as a three-dimensional structures, and we think it's really important, taking into account our previous experience with other viruses. And so the immune determinants are often transient. I told you that the viruses are really not alive, but their proteins, they are in a way dynamic. Sometimes we say they are breathing. They are not literally breathe, but they change their arrangement, like vibrating in the environment. And this helps them to sense the environment and then to bind to the host cell in the right context, enter the host cell or exit the host cell. So the essential epitopes, the essential regions of the viral structure that are really important for the function, they are hidden by these dynamics. So they are exposed only for transient periods of time. And good immunity is often targeting these regions that are kind of hidden by the virus in an evolutionary way. So Virus, on the other hand, on the surface always places, or usually places, some so-called hypervariable regions that are kind of antennas that try to diverse and drive away the immune response. So we always look on the conserved versus hypervariable determinants, and the interplay between them is very important. So what we aim at the first point is to achieve detailed understanding of what are the functional viral antigens. Antigens, those are regions targeted by antibodies and eliciting the immune response. And to put them in place of the life cycle of the virus and understand their importance. And then we would like to understand which natural antibodies in the patient's blood target these particular antigens. And then we would like to know which portion of these antibodies are providing a protection, our ultimate goal, and which ones of those are deleterious, because there are cases where antibodies can actually elicit the deleterious immune response, causing either in overreaction, as I told you, or sometimes just in misreaction. So really understanding the fine-tuning and the determinants is a crucial thing in our thing. So in order to understand the interplay between antibodies and the antigens, we first design and generate recombinantly an assortment of these pieces of the puzzle, viral antigens. And the importance, really crucial importance, is to generate them in the right conformational three-dimensional state, to generate them alone or in the context of the infected cell, or even an assortment of them, like pieces of puzzle assembled together, because sometimes the junction points are by themselves provide an epitope. And so using this assortment of antigens, we apply unique technologies to try to understand what is the composite of the serum and which portions in different patients are reacting with different parts of the viral antigens. And we are looking for the clinical correlates of such reactivity of the antibodies. This will help us to define the protective potential of antibodies using surrogate neutralization assays, looking in vitro in model systems on the interaction between the virus and antibodies and the capacity of the antibodies to prevent, subvert, or sometimes enhance as a deleterious effect viral infection. So then knowing these features of these antibodies will help us to identify the B cells, cells in our body that produce these antibodies, isolate these B cells and subclone using recombinant DNA technology the genes of these that are responsible for generation of antibodies. And this will allow us to produce these antibodies recombinantly in the lab in a well-controlled process, and they can be used directly for the immunotherapy, passive immunotherapy, which is in a way cleaner. And of course, we could prepare cocktails of them. And the next thing would be to characterize the mechanism of their action. How and why do they neutralize and prevent the reactivity? And also those that are deleterious, why are they deleterious? This characterization will involve functional and structural aspects, because really we would like to understand at the level of atomic interactions, where do they bind on the virus and how they interact with the virus. After building a systemic map of these parameters, we can use this knowledge to devise and design new type of vaccines that will only represent these pieces of puzzle that are targeted by those protective antibodies. So in a way, the ideal vaccine would include just those pieces of the virus that elicit protective immune response, and will omit those pieces that are not needed in a way or deleterious. And this approach is called reverse vaccinology. So whereby we are learning from the good immune response in the natural context, after we learn these successful events or successful principles, we can apply them for designing such antigens that would elicit only good and successful immune response. So indeed, often classical approaches lead to a good vaccines, and this is the most usual case, and the ideal vaccine should provide a long-term protection and prevent the spread, protection of the disease, but not only of the disease, also prevent the replication of the virus. But it is not always the case. We still have several pathogens for which we yet unable to generate a good vaccine. For example, HIV or respiratory syncytial virus causing many cases of children respiratory disease annually. Some vaccines are very good, but still they need annual boosting because of the variability of the pathogen, like influenza vaccine. So many research groups are still trying to devise so-called long-term universal influenza vaccine. And some vaccines, like Salk polio vaccine, they prevent the disease, but they are known not to prevent the spread. So for this, usually the combination of Salk and Sabin vaccines are used, while one prevents the disease, the other also prevents the spread. So really formulating the right and the proper vaccine can be a challenge and needs a dedicated research. And in some cases, like for dengue disease or for HIV, the classical approaches are always sometimes fails. And so for this, the reverse vaccinology really takes the part, where this approach of analyzing good instances of immune response from several different patients systematically, looking for the structural epitopes, using the sophisticated structural biology approaches, then helps to map the right epitopes, right regions attacked by neutralizing antibodies, and then use them to generate and design non-natural immunogens, but those that represent only the regions of the virus that elicit protective immune response. So in the last few minutes, I would like just to update you on our recent achievements with the huge effort of several teams. So recent achievements of the CARM team on the immune profiling of the patient sera. And so we have developed an in-house ELISA in collaboration with Adassa Hospital. And you can see here, this is a simple available ELISA test currently done manually in the lab, and it can be upscaled for the robotic platform as well. And so just to start with, we produce two antigens of the virus, the spike and the nuclear capsid, which is a protein inside the virus. And we use commercially available secondary antibodies, chromogenic detection, and it's a quick test for about 19 minutes, a reliable test. And we well characterize conformationally our antigens. And there are several expertises involved and several teams, each dedicated to a specific antigen. And the assay seems to be robust and reproducible. We can determine, you can see here repeats, even not normalized internally, but you can see really low variability. And we receive samples nowadays that are in parallel tested, blindly tested by other commercially available kits. By now we tested more than 230 samples, even I think by nowadays more than 300 samples. And you can see here correlates, and our homemade ELISA performs well. It looks to be a bit more sensitive than commercially available one, and we don't have false positives. You can see a zoom in the comparison. And also we introduced two antigens, and it's quite interesting already. I would just like to zoom in here, so you can see this nuclear protein antigen and the spike antigen. Each one represents blue and this yellow-brown color. So each one is a patient, is just a number for us. We receive it blindly. And you can see here a balanced immune response in this specific patient showing a balanced immune response of antibodies against N, one piece of the puzzle of the virus, and S, the blue one, the other piece of the puzzle. But in another patient, where you can see here, is much stronger immune response against N protein than against S protein. And we can take a different extremity where the response here against S is much stronger than against N. And we are now looking into the functional aspects of this, whether there is a clinical correlate or immunological, other type of correlate, genetic correlate, and all this type of things. And this is quite fascinating also to know which one of those gives better productivity in vivo. Here, just to show you that the pre-COVID cohort really shows no false positive results. It's about 50 samples carefully chosen by the clinical virologists here, and you can see those are positive samples with clinical convalescent phase after clinical disease, and those are negative ones. So, and this is just an example of raw data. We expanded our analysis not only for immunoglobulins type G, but also IgG, IgM, and IgA, while IgA are secretory antibodies, and those are usually acting at the mucosal sites. And this is very important for managing such type of infection that is known to attack the mucosa. And you can clearly see here 3 different samples. One is actually negative, one is intermediate, and one is strongly positive. And you can just by visual observation directly see the results. But this is quantitative because we can use the optical readers to quantify these results. And here this is like a quantitative measurement in dilutions, and you can really see that this is quantitative. For instance, these 2 examples without dilution look quite similar, but when we dilute, we can clearly see that this patient antibodies from this patient are much stronger. And you can see here that the balance between immunoglobulins of different types is different between different patients. And we think understanding this would really help and be essential to understanding the protectivity of the immunity. So we can do it in large scale. Here you can see many patients, and each one of them is analyzed for each type of immunoglobulins for 2 antigens currently, but we are expanding this capacity and trying to put it on the high throughput profile. So just to summarize our current state, we did this simple ELISA that uses well-conformed antigens. We are putting it now on the automation process. We further developed additional conformational immunoassays. And one important and essential point that I would like to stress is that our effort was not to do another commercially available ELISA, but to make it available for public. And we dropped out the price of this ELISA so that we can do this all analysis about 100-fold compared to the currently available commercial ELISA without losing the reproducibility and, of course, the sensitivity. So, of course, what is currently in progress is putting up additional antigens, mutants thereof, including other human coronaviruses to understand the pre-existing immunity in different populations, and trying to understand why some people maybe get the severe disease while others are not. We develop additional immunoassays that would include assays in solutions and more sophisticated high throughput assays for serology. And this will help us to identify individuals with strong and protective immunity, to characterize the antibodies, and then, of course, to clone and isolate B cells, which we are currently in progress of doing. And understanding the mechanism of action of these monoclonal antibodies using structural and functional approaches will help us to design the proper immunotherapy, which will be available immediately. And of course, the further design of proper set of antigens that can be used for formulating subunit recombinant vaccines. And so thank you for joining this webinar. Now I'll be happy to take some questions.
Exactly. Thank you very much. So we now open the question and answer time. If you have any question, you can raise your hand. or you can write it on the chat. So let's wait a few minutes. Okay, we have Samuel Zalipski. Samuel, you can open your microphone and speak.
Yes, hi.
Hello.
So I understand that most healthy individuals have antibodies against human coronaviruses because of their exposure to common cold. Is it correct to assume that there is fair degree of homology between the human coronaviruses and the current virus that causes this epidemic? And then what are the implications for some people having very light disease? Is it possible to assume that the presence of these antibodies protect those individuals? And also, what are the implications for analytical purposes? Because these presence of these antibodies against human coronaviruses, obviously they have common epitopes with SARS-CoV-2. So those are my questions. Thank you.
Thank you, Samuel. So really exciting questions, and of course this is something that we believe needs to be addressed systematically. There is a certain degree of homology, and the question is which type of epitopes are cross-reactive and they do elicit antibody response for which specific human common cold coronavirus. And what we are doing in order to address these questions, we are generating the expanded panel of antigens, recombinant antigens, representing different regions of human common cold coronaviruses and of SARS-CoV-2. And by systematic analysis of this is actually undergoing currently in our labs, in our team, current team, and we put them all one by one in immunoassays, and then we can compare a given individual toward these reactivities, and only then we can really give an answer for these questions, which are really essential to understand what is the degree of reactivity of antibodies, cross-reactivities, and of course, what is the degree of protection, cross-protection, which is actually the main, if I understand correctly, the main essence of the question, whether there is a cross-protection or not, and toward which coronaviruses, and even more than that, toward which epitopes, specific regions, toward which antigens. Mel, I think you are in mute. I cannot hear you.
Sorry. Okay, now we have a question from Anatya Nai.
I hope you can hear me. Hi, Alex.
Hi, Anat. Hi.
Great to see you after almost 20 years. And thank you for the exciting seminar. I was wondering for your patient ELISA assays, the ones that do not react with the two antigens, that they're almost like healthy patients, they don't have any immune response. What do you suggest where they fall in the, in the assay?
Yes, so in a way, the dynamic range of this ELISA is quite large. So we have these outliers which have a very strong immune response, and we also can see very moderate reactivities. But some of the patients are just negative. So this is not only patients in the cohort we studied. Those are not only patients hospitalized for COVID. Some of them are just randomly taken, so it's a blind cohort. So except for these 50, I can show, except for these 50 patients that I showed you here, you see this pre-COVID cohort where we know they are negative. The others are just, some of them are positive here, and some of them are positive but didn't yet elicit strong antibody response because they are too close to the phase. Mm-hmm. And also maybe they elicit antibodies against different parts of the antigens. What we are currently using is only the most antigenic part of the spike and most antigenic part of the N protein. But currently we are expanding this panel to include further parts, and then maybe we can see other reactivities. That's exactly the aim, to really expand the panel.
Thank you very much.
Oh.
Thank you. Now we have a question from Manoj. I hope I said the name right. Manoj.
Yeah. Hi, Alex.
Hi.
It was great to learn many things about the coronavirus. My question is not basically pertaining to the innate response. My general question is like in earlier slides you have shown that this coronavirus has the spike proteins, and you was talking about this conformational change, like there is a breathing of this protein or these spike structured proteins. So is there can be any relation that this can help in determining the spectral properties of this these spike proteins? Am I right?
So you mean, yes, one of the things we are currently generating is stabilizing this conformational assortment. So we generate recombinant proteins of this versions of the spike that would be more breathing or more fluctuating or less fluctuating, more stabilized ones, and even sub portions of this spike, so that if we put them on the panel ELISA, we can really measure the reactivities between each of them, and even in some cases we can use them in competitive ELISA, whereby we can, for instance, use one antigen and put another one to compete or to outcompete, and then by this we can map the exact epitopic regions or at least narrow them down. That's the idea.
Okay.
Okay.
So we have now a question from Nathan Ronin.
Hello, Alex.
Hello.
Hi, thank you very much for the lecture. I would like to ask, at the beginning of the lecture, you talked about some kinds of illnesses that are related somehow to coronavirus and are like genetically similar. I would like to know, have you tried comparing the antibodies that you've seen that are working in ELISA on the coronavirus on those, like comparing them with the antibodies that are produced from these kinds of illnesses that we talked about earlier that we actually know more about?
So just to refine the question, you are asking about whether we try to see whether antibodies from COVID patients can neutralize the other common cold coronaviruses or vice versa?
Yes, like if there are similarities between them.
Yes, this is one of the things we are addressing just nowadays, also by cross-reactivity, but also by functional aspects, like whether they can inhibit really the propagation of the virus. This is one of our current goals.
Thank you very much.
Okay, so we have now from the chat, Anna Wenger. She's asking what is the error rate of the test?
So the test is currently in the final stages of validation in tight collaboration with the clinical virology, and currently it behaves very well. I don't have yet numbers, but it really behaves similar to the commercial alternatives, Euroimmune, and currently we are testing it against Abbott also, Euroimmune and Diasorin also. So we are comparing with this large cohort against currently commercially available kits used in clinical diagnostics, and meanwhile we get a very positive feedback. We do it blindly, so when you look on the errors of the kit, you can describe it in false positives and also in false negatives. So we seem to be more sensitive than some of the currently available kits. And for false negatives, we don't see yet false negatives, but we are expanding the cohort. And also we are looking for pre-COVID cohorts to better evaluate the cross-reactivities. That's the current state, but it's behaving quite well.
Okay, we have another question from the chat from Yvonne Engel. How effective would the vaccine be for the elderly people with a compromised immune system or people with an autoimmune disorders?
So yes, it's a bit maybe too early to speculate on that in my view, but of course here I would also think on the passive immunotherapy option, where you can with people who cannot really elicit the proper immune response or that cannot be vaccinated for any reasons of the failure. Such patients, as far as I know, though I'm not a clinician, they can be treated with immunoglobulins, either enriched immunoglobulins from patient sera, or what is even better, by recombinant monoclonal antibodies or cocktails thereof. That's the approach we envision.
Okay, we have another question from the chat. What is your view, Alex's view, as to why the virus outbreak in Africa has been less than expected?
This is mostly, I can raise several thoughts that are not necessarily mine, but are there around. So one thought could be just the genetic background, or another thought could be pre-existing immunities. Another thought could be also the surveillance, because really surveillance is very important, as we can see, and the numbers sometimes follow the amount of tested cases. So I think currently is a bit too speculative, but also maybe the pre-existing immunity against other coronaviruses could be a factor, specific coronaviruses like raised before, or maybe other viral or non-viral infections that could also have an impact, or maybe genetics. So this, I think it's too early because we need more epidemiological surveillance studies. And one of the thoughts behind available tools to do such surveillance was to develop something that is available also not only in developed countries, but also in developing countries. That's one of the thoughts, to make something really available for the public.
Okay, so we have time for one more question. If you have it, we can wait a few seconds. Okay, so I think we are done with the question. Thank you very much, Professor Rouvinski. It was great and very interesting as usual. And you will all receive the recording tomorrow, and hope to see you in the next webinar on Thursday. Thank you to everybody.
Thank you. Thank you. Thank you.